Welcome. In the last video, we have learned about the blotting techniques and also we have seen about the southern blot. Now, today we are going to learn about northern blot. Northern blot technique, it has been invented by the Alvin, and this method is named according to the name of the southern blot. As it is a southern, accordingly that it is a northern. That is, there is a not any significance for the northern. That is, the northern, it is not an inventor or anything. Okay, it is just a southern and next one, it is a northern. So northern blot, it is also going to be called as a northern hybridization. Okay. Now, when you look at the northern blot, it is used for the detection of the specific RNA, and also it is going to be used for identification of the expression of the specific gene by measuring the amount of the RNA. Now, when you look at the steps for the northern blotting, first you have to go for the isolation of the cells, and then you have to go for the lysis of the cells and the membrane disruption. And in the cell, there is the presence of the ribonucleases. Its activity, it has to be inhibited because you want the stable RNA. Then you have to go for the deproteinization, and then you are going to isolate the intact RNA. And after the isolation of the RNA, the most important step that you have to perform, it is a denaturation of the RNA to resolve the secondary structures. Now, the northern blot or the northern hybridization, this technique, it is allowing the detection of the RNA by the hybridization with the probes. The probe, it may be the DNA or the RNA. That is, there has to be the binding between the RNA and the probe. And for that purpose, there should not be presence of any secondary structures. Okay, that is the RNA molecule, it should be, okay, it should be in a single stranded form. That's why denaturation of the RNA to resolve the secondary structures, it is very important. Now, when you look at the procedure, first you have to go for the separation of the RNA, and here the gels, it may be the denaturing gels because you have to resolve the secondary structures. Then you are going for the electroblotting, and when you are using the membrane, it is a DBM paper. Okay, nylon paper, here the DBM paper, it is used, which is derived from the Whatman filter paper. Nowadays, there is a availability of, okay, nowadays there is the availability of the nylon membranes, which are allowing the binding of the RNA. Now here, when you go for the electroblotting, here the cassette, it is going to be made, and in this cassette there is a presence of the sponge, filter paper, membrane. On the membrane there is a placement of the gel, and again filters and a sponge. And this cassette, it is made, and it is kept into the electroblotting chamber, and the electric current, it is allowed, and that electric current, it is allowing the transfer of the transfer of the RNA bands over the membrane. Okay, and after this transfer, then the next step, it is a pre-hybridization step, and in pre-hybridization step, the blocking, it is going to be done on the membrane. The spaces where there is no presence of the RNA bands, that space, it has to be blocked because there should not be any non-specific binding of the probes. That's why the blocking, it is done, and that blocking, it is done with the help of the BSA or the blocking agents. Now, the next step, it is a hybridization. After pre-hybridization, then the hybridization, it is done. In the hybridization, you are adding the radio-labeled DNA or the RNA molecules. Now, some of the time nowadays, the fluorescent molecules or the fluorescent labeled probes, they are going to be used. And this radio labeled DNA or RNA or the fluorescent labeled DNA or RNA, it is what? It is the probe. Okay, and it is allowed to get hybridized with the RNA. That is, wherever there is a complementarity between the probe and the RNA, the probe, it is going to bound with whom? It is going to bound with the RNA. And there are some of the probes which are not going to be bound. Such a probes which are unbound one, it has to be washed out, and this, it is going to be washed by the stringent washing. In the stringent washing, only the specific probes, they are allowed to get bound, and the non-specific or the probes which are unbound or which are the extra one, they are going to be removed. And after that, okay, after stringent washing, then you are going for the detection. And detection, it is by the autoradiography when the probes, they are the radio labeled. Okay, in this way, the procedure of the northern blotting, it is going to be done. I think you may have cleared the idea about the northern blot. If you like my video, please subscribe my channel and also share my video with your friends so the knowledge it is going to spread. Thank you.